Hello, voice audible to you? Hello, is it clear?
Yeah.
Right. So, are you able to see this slide PPT reproduction in bacteria?
Yes, sir.
Right, reproduction in bacteria. Is it clear? PPT slide clear?
Yes, sir.
Right, actually, in the last class, we completed about bacteria structure, right? Hello,
Yes, sir.
just wait 1 minute. In bacteria cell structure, we completed about bacterial cell component, cell wall, bacteria cell capsule, plasma membrane, nucleus, pili, fimbriae, plasmids. We discussed all about that. Now today another important topic about bacteria, bacterial reproduction. The reproduction types we discussed in reproduction type, vegetative and asexual and sexual reproduction. We are going to discuss today. The first in vegetative reproduction. The vegetative reproduction, the most common method is binary fission. Of course, there are some different type of vegetative reproduction also there. But in this, the most important method and common method is binary fission. Binary fission means bi means two. One bacteria becomes two. That is called binary fission. So it is the commonest type of reproduction under favorable condition. So in which divided one bacteria into two similar daughter cells. So during the process, first the bacterial chromosomes get detached to cell membrane and replicate to the bacterial chromosome. Actually, what happened before the replication of chromosome, the first the bacteria cell. Here, let me take the pen. The first step, the bacteria cell somewhat elongate, somewhat extend in size. The later, in third step, small construction formed between the cell, the small construction like this. The meantime, this chromatid or bacteria chromosome undergoes replication. Replication means one DNA strands become another DNA. That means one becomes two strands. This is called replication process. After replication process, along with the cytoplasm, this the bacterial genome also separated. Bacterial genome also separated. After that, the small constriction, the extend to each other, opposite direction, finally the septa form. Now the finally these are detached each other and divided into two daughter cells. The first during the process, first bacterial chromosome get detached to cell membrane and replicate like this. And replication means it becomes double. As the cell enlarges, the daughter chromosome gets separated. Already told you, a cross wall formed between the separated daughter chromosome. This is the cross wall. This is the cross wall. Now this cross wall divides the daughter cell into two. So this is called as a binary fission. And next one, just wait. After binary fission, Another one is endospore formation. Another reproduction type is endospore formation. Another one is endospore formation. This endospore formation by asexual reproduction. Binary fission, one method of the vegetative method, because the entire bacteria body divided into two. That is considered as a vegetative method. And in asexual method, endospore formation. So asexual reproduction takes place by spores first, whether it is exospore or endospore, whatever it may be, by spores. And sexual reproduction takes place by gamete fusion. Remember it. This is the general statement. So vegetative reproduction, any vegetative part involved to reproduction. In asexual reproduction, involved by spore formation, and sexual reproduction by gamete formation. Is it clear? So first in vegetative method, of course there are different type of methods there, but not important one. But here the most important one, binary fission, that is completed. Now the part of the asexual reproduction, spore formation, that is endospore formation. Now let's discuss endospore formation. Endo means inside. Hello? Let's take pen. Endospore formation. Slide clear? Endospore formation. Are you able to see the slide?
Yes, sir.
Yes, sir.
Right. So this is formation during asexual reproduction. Here the endospore formation. Endospore are resting spores formed in some gram-positive bacteria. Example, Bacillus, Clostridium. Sorry, actually these are the formed during right. Endospores are the another name is resting spore. They are formed in gram-positive bacterium, Bacillus and Clostridium. As they are resting spore, they are formed only during unfavorable conditions, but not favorable. When the conditions are not clear, then only these spores are formed. So they are formed within the cell. Formed within the cell. During this process, the part of the protoplast becomes concentrated around the chromosome. That is, the entire protoplasm, including chromosome, rounds up, and it is covered by and synthesized by new layer, the hard resistance wall secreted around it. So rest of the bacterial cell degenerate during unfavorable. What is first unfavorable condition? What is first unfavorable condition? What is unfavorable condition?
Not
Not
suitable.
suitable.
Right. Unfavorable condition, not suitable to the growth of the bacteria. So what are the situation? What are the not suitable situation? Maybe high temperature, low humidity. These are the unfavorable conditions. So, and also lack of scarcity of nutrition. These are unfavorable conditions. In this situation, what happens? Just wait. Right. So this is endospore formation. Where it is? Why it is not completed? Just wait. So this is endospore formation, right? Right, endospore formation. The first, this is the bacterial cell. In bacterial cell, this is the protoplasm. What happened during unfavorable condition? Inside the bacteria, the entire protoplasm round up and is synthesized by new layers. New layer. But during unfavorable condition, when water scarcity, nutrition scarcity, high temperature, this bacterial cell destroyed. Bacterial cell destroyed except the endosperm, as it covered by thick layer inside the content of the protoplast, not damaged because of thick layer. Now it is withstand to the unfavorable condition. This layer, this endospore, withstand and survive for unfavorable condition. When the favorable condition reoccur, now it is developed into the new bacteria. Developed into new bacteria. So this is called endospore. This is called spore, which is formed inside the bacterial cell. That's why it is called as a endospore. Is it clear? Binary
Yes.
fission and endospore formation. Simple endospore formation inside the bacterial cell by the roundup of the protoplast, including genetic material. Right, in protoplast itself, all the cell organelles and genetic materials also included. So that's why after formation, the spore from protoplast or protoplasm, it is covered by new layers, thick layers. As it thick layers, it is survive during unfavorable condition, but not the rest of the bacterial cell. The lack of scarcity of water and food material, high temperature, the bacteria cell would be die, but not the endospore. When the conditions reoccur, now it is developed into new bacterial cell. Right? Next one. And conidia formation. The another asexual reproduction method is conidia. First one is endospore formation, and second one is conidia formation. In some bacteria, such as Streptomyces, considered as a true gram-positive. Here the gram-positive Streptomyces, the gram-positive bacteria, and the endospore formation also in gram-positive bacteria. So from the bacteria, conidium are formed like a chain, conidia chain. I will show the diagram later. Each conidia generates to gives rise to new bacteria thallus. If you see the diagram, right. For example, it is for rhizobium, right. For example, this is bacterium. From bacteria cells, conidia chains like this one, conidium. Conidia are formed exogenous, but not endo. Exogenously, conidia are formed like this. Conidia chain formed. This is the bacteria cell. This is the bacteria cell. On the bacteria cell, the conidia develop like a chain like this. Due to any reason, these are the conidia. Yeah, these are the conidia. After formation, this conidia liberated out and germinated into new bacteria cell. So this is about conidia formation. So first, asexual reproduction method, endospore formation. Second one is conidia formation. Next one, sexual reproduction. So already told you what is sexual reproduction. What is sexual reproduction?
Reproduction through gametes.
Through gametes, right. But what happened in the bacteria? In bacteria, any gamete formation, any sex organ present in bacteria?
No, sir.
Like we discussed in first class, algae. In algae, what are the reproductive organ? Algal members. Algae reproductive organ, male one antheridium or male gametangium, female one female gametangium or archegonium.
Yes.
But in bacteria, as it is prokaryote and simple structure, unicell, that means entire cell made up of single cell. That's why there is no special reproductive organs in bacteria. That means there is no male gametangium, female gametangium, or antheridium, oogonium,
Yes.
and also there is no gametes also. No gamete formation. In bacteria, no male gamete formation and female gamete formation. Then how the sexual reproduction takes place?
Through cells.
So another important concept of the sexual reproduction is here. What happens in sexual reproduction? First of all, for example, this is the gamete. This is also gamete. Here, let me change color. For example, this is the plus gamete.
Sir,
Just
in question, if they give bacterial transduction or if they give simply transduction, for both the questions, this answer is same only, no, sir?
yes, same
Okay, sir.
answer, same transduction. That is the one type of sexual reproduction in bacteria.
Okay, sir. Thank you, sir.
Okay. So the plus, what is plus and yellow color minus. So now the one is considered as a male one. This is male gamete, for example. This is female gamete. Now the both gamete fusion. This is plus, nothing but here the plus is the genetic material, blue color, minus is genetic material, orange color. What happened later after formation zygote? Let's change the color. For example, this is the zygote 2n. In 2n zygote, what happened? The plus, sorry, 2n plus color plus blue color minus orange color. Both are present, and these are exchange their genes. The plus gene and minus genes both are exchanged. This plus to minus, minus to plus. These are both are exchanged. So this is called genetic recombination. This is called genetic recombination. So that's why here what I'm going to tell, even though there is no gamete formation in bacteria, but another important aspect of the sexual reproduction, recombination takes place. These recombinations occurs during the sexual reproduction in bacteria, right? So there are three methods of genetic recombination. The recombination nothing but exchange of the genes, plus to minus, minus to plus. Here the mainly three methods. One is transformation, conjugation, transduction. Remember, transformation, conjugation, and transduction. Here the transformation means the genetic material transferred without any media. The genetic material transferred. The conjugation also is the gene material transformed from the one bacteria to another bacteria. Transduction also genetic material transfer to one bacteria to another bacterium with help of the virus. I will explain clearly. Here the first, let's see the transformation. In transformation, just wait. transformation. So transformation first studied by Griffith. Let's change color. The first studied by Griffith and Diplococcus pneumonia bacteria. Diplococcus pneumonia bacteria. In transformation, DNA from the donor cell initiate irritable change in recipient cells. Remember this word. The transformation involves only one trait, so the several may be acquired independently in this way. Let's see the transformation process. The first of all, it is first discovered by Griffith in Diplococcus pneumonia, right? If the genetic transfer in bacteria also occurs by transformation, what happens in which the DNA molecules of the donor cell, for example, is there given any? Just wait. Diagrams. Right. Bacteria transformation. It's not clear. So here the bacteria transformation, the two bacteria. Bacterium with gene per capsule. This is the heat killed bacteria disintegrated and release the DNA into surroundings. This is the normal bacteria. Okay. This is heat killed bacteria. Heat killed bacteria. And the what happened? The bacterium gene, this gene transfer into the heat killed bacteria. Heat killed bacteria. Later what happened? Actually, this diagram not clear. I will draw. Right. Whiteboard clear? Whiteboard. Are you able to see whiteboard?
Yes.
Right.
Mm.
Whiteboard. Right. Bacteria. Capsulated smooth cell virulent. One is virulent bacteria. Sophia. Sophia, don't draw. So, okay. Don't draw diagram. So there are here the given different type of bacteria. One is virulent bacteria This is A, and another one is Ambrine. Ambrine, don't draw. B. This is non-virulent bacteria. Galaxy A30, remove it.
Friends, please don't do unnecessary things on the board. Sir is trying to explain us. Don't waste Sir's time.
Hello. So B cell non-virulent. B cell non-virulent. Now in third step, what happened? This virulent killed by heat. Virulent bacteria by heat. Heat kill. Virulent bacteria. Heat killed virulent. That means when you heat, this bacteria killed. Now next this bacteria, that means non-virulent bacteria gene, B gene, mixed with virulent bacteria. What is the tendency of virulent bacteria? Actually, this virulent bacteria cause the disease. It has the pathogen character. It cause the disease to the others. Non-virulent bacteria never harm to the host. Does not produce any toxins. Does not cause any host. It is not a pathogenic character. What happened here? Now, the scientist Griffith, in his experiment, this virulent tendency of causing disease, the virulent bacteria killed by heat. What happened during this heating? The A gene automatically lost their tendency. But when the mixed with the gene B, what happened? After the both genes are mixed, automatically this bacteria turns into virulent. How it is possible? That means non-virulent bacteria turns into virulent bacteria.
How?
Actually, this bacteria does not cause any disease. But in what happened next time, the both, that means heat killed bacteria and non-virulent bacteria both are mixed in a device. Automatically, what happened next? Immediately this that's a non-virulent bacteria turns into virulent bacteria. Virulent bacteria. That means it has the tendency of the cause disease. But before mixing, there is no tendency to cause disease. What you understand in this experiment, in this Griffith experiment? Hello? Please
Yes.
remember this diagram. First, he selected the two bacteria, right? One is A virulent bacteria, another one B is non-virulent bacteria.
Non-virulent bacteria.
Yes, what is the tendency? The characteristic nature of the A virulent bacteria, if individually A virulent bacteria inject to the mice, virulent bacteria. Separately, this virulent bacteria injected to the mice, rat. What happened? Due to the virulent nature, the mice would be die or alive.
Diet, sir.
Die. The mice die. When you separated non-virulent
Non-virulent
bacteria,
bacteria, it
now
survives.
non-virulent bacteria injected to the mice survive.
The mice will
It
survive.
works live. But later, he injected the heat-killed bacteria. Only heat-killed. Only heat-killed. Heat-killed. That means give the symbol as a heat-killed
Virulent.
into. That means heat-killed. Now the only heat-killed injected the mice. Now what happened? Heat-killed injected the mice. What happened?
Mice survive.
Mice survive, not killed.
Survive. Not killed.
But now for fourth step, this heat killed bacteria, in fourth step, heat killed bacteria That means heat killed bacteria mixing with non-virulent bacteria. Non-virulent bacteria. Mixing with non-virulent bacteria. Now, automatically, due to the gene transfer, automatically it has get the tendency of like a virulent. Now this bacteria injected into the mice. But now this time the mice would be die. This time mice would be die. Right? So what happened during this? Here the changing the gene, that means B genes enter into the virulent. Automatically the virulents get active. Actually, when you eat, it is inactive. When the B gene enter into this, it is also active, and they have the tendency of virulent, like a virulent bacteria. Now in this situation, the mice will die. Okay, is it clear? Hello?
Yes, sir.
Right. Now, so transformation clear. Now another one is,
Sir, excuse me, sir.
uh,
Sir, if the question asks Griffith's experiment, so we have to explain this only.
yes, Griffith experiment. The Griffith experiment or write about bacteria
Or
transformation.
bacterial transformation also Griffith experiment only, sir.
Yes, both are same. Griffith
Yes.
experiment, Griffith experiment or bacteria transformation. If they ask a short answer, you can write about this.
Yes, sir.
And next one, conjugation. Completed, endosper formation, transduction. Conjugation. So first of all, it is proposed by, let me change. It is first reported by Ledenberg and Tatum. Remember the scientist? Ledenberg and Tatum in E. coli bacteria, Escherichia coli bacteria. So how the genetic material transform in this? Here the given two type of cell, HFR cells and normal cell. The cell-to-cell union occurs between the two bacterial cells, and genetic material or bacterial genome or DNA of one bacterial cell goes to the another cell through the conjugation tube. This is important. Through the conjugation tube, this tube, which is formed by sex pili. Already we discussed the pili in bacterial cell structure. As a result, recipient of cell possesses duplicate genes for a number of characters as seen in the Escherichia coli in inhibitor of human intestine. So for first step, HFR cell contains genes. Here, this is HFR cell that contains genes that allow them to transfer. What is this here? Allow them to transfer some or all other chromosomes to another cell. In HFR cell's chromosome, replication takes place here. Actually, in the HFR cell, plasmid replication takes place. The replication strand, another replication strand transfer into the another cell. For example, this HFR cell, F cell means it has F plasmid. The F plasmid undergoes replication. Replication that means double. After replication, one strand transfer to the another one. So here the plasmid as it is, and here the another plasmid strand migrated to this. Now what happened? This is called F plasmid, F plus. But in normal cell, is there F plus plasmid present or absent? Present now. In normal cell, F plus plasmid present or absent? Before conjugation, absent. So it is considered as a minus, right? Now F plus plasmid, what happens? The F plus plasmid undergoes to replication process. That means they become one strand, become replication, and replicated strand transfer into the F minus. What happened in F minus normal cell? Normal cell also plasmids existed. So here the HFR cell, here the HFR cell. Here is the plasmid and here is plasmid. Now what happened finally? The both are turns into F plus cells. F plus cells. Now the both are turns into F plus cells. So like this, the genetic material takes place. Is it clear? Hello?
Yes, sir.
Right. So conjugation process also clear. Actually, get the conjugation tube. Actually, this is 6 pili. First, the 6 pili F plus cells get attached with the normal cell. There is no 6 pili. With help of this pili, attached to the another normal bacteria. Later, this pili like a formation as a tube, conjugation tube. Through the conjugation tube, the genetic material transfers. Right?
Yes, sir.
But remember the scientist names. Griffith scientist transformation, and Griffith bacteria pneumonia, and conjugation Lidenberg and Tata
Yeah.
bacteria Escherichia coli. Right? Any doubt in this?
No, sir.
Next one, another important transduction. Transduction. Right, transduction. First reported by Ginder and Lidenberg in Salmonella typhi. Salmonella typhi. Important scientist name Ginder, Lidenberg. Lidenberg tatum conjugation. Ginder and Lidenberg transduction. In this method, genetic material of one bacteria cell goes to another bacteria cell by agency of bacteriophage. This is important. By bacteriophage. Bacteriophage. The bacteriophage, that means virus infecting bacteria. Let's see in diagram about transduction. Right. So I already told you the transduction takes place, genetic material from one bacteria. Let's take color. For example, this is the A bacteria. This is the B bacteria. Now the genetic material transferred to the A bacteria to the B bacteria. Right? Let's see how this is formed. The first of all, I already told you. So now this is bacteriophage. Already you know the structure of bacteriophage, like a tadpole shape like this. This is bacteriophage. This is host bacteria. This is bacterial chromosome, a bacterial chromosome. This when the phage bacteria injects its DNA. During adhesion process, the bacteriophage attached to the bacterial host cell. Later the bacteriophage, that means virus genome, this genome enter into the bacteria cell. This is phage DNA or virus DNA. What happened after enter the virus DNA? Immediately the bacterial chromosome disintegrate like this, break into pieces, disintegrate. Now the phage enzyme degrades or degrade the host DNA. Now the phase with donor DNA transduction phase, that means simple. After breaks into pieces, each one of the bacteria, each one of the genome act as a template to formation of the RNA strand, RNA form. After RNA formation, on the RNA, proteins are formed. These protein utilized for the phase variant. Phase variants means like egg formation, egg made up of protein. head formation, collar formation, tail formation. They are all formation by proteins. While the protein form, here the genome also settled at center. The bacterial genome also settled at center. Right? While this formation, this genome, bacterial genome, a bit of phase DNA, let's change color, bit of phase DNA also attached to it. Now that this bacterial genome, red color, while the phase formation, the bit of phase DNA also attached to the bacterial gene. Now what happened? Now in this bacterial phase, two DNAs are present. Phase DNA as is, with help of phase DNA, the bacterial DNA also present. Bacterial DNA also present in, let's change color, bacterial DNA, for example, bacterial DNA is red color, yes, bacterial DNA also present. In phase, bacterial DNA also present. Now what happened? Finally, the lysis takes place. I will explain lytic cycle later. Lysis takes place. Lysis means due to the lysozyme enzyme, the bacterial cell wall ruptured. As a result, these phases liberated or released out of the cell. Now this phase DNA or bacterial phase infected to another bacteria. Now the same process means when enter the viral DNA or phase DNA, automatically the bacterial DNA disintegrate. Now this DNA mixed with bacterial DNA. Already in this phase DNA, what is present? In this phase DNA, not only phase, and also it has the viral DNA, bacterial DNA also present. So bacterial DNA with viral DNA. Now this DNA fused with another bacteria. So like this, here the conjugation, recombination takes place. Simple, don't get confused here. Don't get confused, first of all. So this is bacteria genome. After virus genome enter, actually what happened? This virus genome disintegrate. Disintegrate means when it is attached to the bacterial DNA, automatically it is break into pieces. Now the bit of the phase DNA mixed with this bacterial phase, mixed with the host bacterial DNA. After that, protein synthesis, these protein variants are synthesizing, utilizing or formation of the bacteriophage parts such as the tail, head, collar, tail plate, tail fibers, there are. So after formation this bacteriophage, in this bacteriophage, phase DNA and A bacterial DNA present. When now it is after lysis, it is liberated out and infect another bacteria. Now what happened? Already inside the phase DNA, A bacteria DNA present. Already A present. Now this A bacteria DNA fused with B bacteria DNA. This is B bacteria fused with B bacteria DNA. Like this, the conjugation takes place. Is it clear? Transaction clear or any doubt in this? Hello?
Clear, sir.
Right. Now, next one. The virus replication. Another important virus replication. Just now we discussed about lysogenesis process like that. The virus reproduction, another important one. So in this unit, bacteria complete bacteria cell structure and bacteria reproduction complete. Now in virus, and actually in our book, what are the rest of the parts of the bacteria? Bacteria after reproduction, I think you are given bacteria economic importance. Classification not important, but economic importance important. Economic importance of bacteria.
Okay.
There is no point to explain because it is the generalized. Already you must have read since the from the 8th class. Role in agriculture, agriculture role, industrial role, plant role.
In
That
medicine.
means right. So these are the some general topics. So I don't waste your time and my time.
Yes.
Should I explain the economic importance?
No, sir, it is understandable.
Economic importance, I suggest you one thing. In economic importance, first of all, you just go through the headings. First, role in agriculture. How it is utilized for agriculture. This bacteria utilized for the decaying organic substances can improve the fertility of the soil. How it is improve the fertility of soil? Can you give an example? Hello? How the bacteria improve fertility of the soil?
Nit
Ah,
by nitrifying nitrogen.
by nitrogen fixing bacteria. Nitrogen fixation takes place in which plants? Nitrogen fixation. Rhizobium bacterium, nitrogen fixation in plants. Which plants? Which family plants?
Rhizo.
Leguminaceae family. In leguminaceae family.
Nodule.
Right, root nodule formation. Like that, there are different ammonifying bacteria, nitrifying bacteria there. And third one given nitrogen fixation bacteria in leguminaceae family by rhizobium. And second one is heading is role in industry, medicine. Industry, give some example, general, in your daily life example. I am asking you daily life example, how the bacteria are utilized to you in your daily life.
Curd
That is outcome.
formation, sir.
Right. That is outcome based question. Now,
Lactobacillus bacteria. Curd formation.
cord formation. Right. Another important
Yeast. That
is not a
dosa.
bacteria. Is not a
Sorry,
bacteria.
dosa batter.
Okay. And preparation of alcohol also. Alcohol formation by Clostridium estobulum bacteria. Preparation of vinegar. Do you know vinegar?
Cheese.
Yes.
Cheese.
Cheese, vinegar formation, estobacter. Tea, coffee, tobacco, hemp fiber, extraction of the fiber from the hemp.
Sir, in tea coffee formation also bacteria helps, sir.
Yes. That means flavoring.
Flavoring. Okay.
Yes. So, these are the useful bacteria. Is there any harmful bacteria? In economic importance means you can write the both useful and harmful. If they write example only write about useful bacteria, you just write about agriculture and medicine and industrial field. Only they ask about the harmful bacteria, you can write the plant disease and animal disease. Now tell me the harmful aspect. Animal pathogen bacteria given, plant pathogen bacteria given, food destroying bacteria given, soil destroying bacteria also given. Now, some tell me the animal pathogenic bacteria, that means in just give me the human disease of bacteria. I am asking just human diseases of bacteria. Human disease of bacteria. Give me example. This type of question we are going to ask outcome based question, last question. Actually, in outcome based in previous paper, objective. Instead of objective papers, 20 marks given as the outcome based. If I ask write some of the harmful bacteria in human beings.
Tuberculosis.
What is that? Of course, in our book not given some of the disease.
Yes, sir.
Even though you can write the human diseases. Now tell me, already you must have read, you must have learnt in previous year, in previous classes. Now tell me one or two examples.
Tuberculosis. Tetanus.
Yes, tetanus. Tetanus bacteria.
Diphtheria,
Next,
sir.
diphtheria. Okay. Next one. Clostridium tetani causing tetanus. Okay.
Cholera.
Cholera. Vibrio cholera bacteria cause cholera. And the most common fever.
Typhoid.
Typhoid. Typhoid by Salmonella typhi.
Salmonella typhi.
So these are the some of the bacterial disease.
In human
And
being.
like that, can you tell the plant diseases of bacteria?
Citrus canker.
Citrus canker bacteria, blight of rice, wilt of cotton. Some of these are the bacteria
Potato
diseases.
light blight tumors.
Potato rot,
Potato.
not late blight. Potato rot.
Wheat rust.
No. Wheat rust is the fungi disease.
Fungi. Okay, it is called
Fungi disease. That's not bacteria disease.
Okay.
So these are the harmful. And food destroying. If you leave the food above the 24 hours, what happens? Not only food, any hittable things.
Right.
What happens after 24
It
hours?
will get spoiled.
Spoiled. The which bacteria involved in this? Whatever it may be, the food stuff, including meat and milk, vegetable, fruits, any type of food gets spoiled by bacteria. The name of the bacteria, Clostridium botulinum.
Clostridium.
The cause food poisoning. Right, another soil fertility also destroying with help of the bacillus denitrification. Nitrification means nitrates formation. Denitrification means remove the nitrates, remove the nitrate from the soil. What happens if the nitrate remove from the soil?
Fertility loses.
Right, the loss fertility, that means the loss of
Loss of
nitrogen minerals. Right, and here the common plant diseases given in chart, page number 63. Bacterial diseases and factors inducted spread, crop infected and symptoms. But here the most important thing, only type of bacteria disease and symptoms. This much enough. Is it clear?
Yes, sir.
Yes, sir.
Right. So if you take virus, in virus already we discussed virus structures while we discussing about the bacteria structure. In the bacteria clear structure there, but when it comes to the virus there is no clear structure. Only in virus already told you the virion protein coat and nucleic acid there. So that is the very simple structure. And also virus is have the both tendency, both character, living nature and unliving nature. Please be silent. Allow. Please be silent. Inside the host it shows the living. Outside the host non-living. So that is the characteristic nature of the virus. And here the virus, another important one is replication. In replication, let's change color. Just it complete in 5 minutes. The first
Sir,
step,
bacterial growth curve and bacteria growth period means both are same question, sir.
bacteria growth
Curves, bacteria growth period.
Growth curve, growth period, both are same.
Okay, sir. Thank you, sir.
Growth curve in page number 51, if you see, this is also important question. Bacteria growth curve or growth period. There are how many stages clearly given here? The 3 stages: log phase, Eliage log phase, and LOG log phase and stationary phase. Another final one is decline phase, fourth phase also there. So what happened in log phase? It just focus on the graph. For example, log phase initial period. This is initial phase. One is number of cells. This row number of cells. And here log phase initial period, the time required for adoption. Maximum size cell obtained toward the log phase. That means what happened during log phase? While you increasing time, almost stable. When log phase, automatically the number increase. This is the symbol, number increase. And again stable. The finally what happened? Decline. So this is the growth of the bacteria. First stable. This is called LAG log. Next LOG log phase. Rapid increase number. Please be silent. Please be silent. Now stationary. Hello. Right. Now after that decline stage, completely decline. So here the growth curve and growth period both are same. And another important nutrition and growth. So growth completed, right? Nutrition, should I explain? Nutrition. So simple in nutrition of bacteria, chemotropes, autotrophs, phototropes. What is phototropes? Photo means
Light.
light. They
Bacteria
can
which
prepare,
sunlight.
right? Right. Some of the bacteria can prepare their food material with the light. Chemotropes means they can prepare with help of chemicals.
Chemicals.
Heterotrophs,
Dependent
dependent others.
Or
In heterotrophs in bacteria, maybe saprophytes, maybe parasites. What is saprophytes? What is saprophytes?
Dead and
soil.
Death and decay. What is parasites?
parasites.
Depend on others.
Depend on other living host. If death and decay, that is saprophytes. If the bacteria, the live on other living host, that is called parasites. So this is the nutrition means how the food material getting of the bacteria. So the bacteria may be some of the bacteria getting by autotrophic nature. That means photoautotrophic bacteria separated and chemoautotrophic bacteria separated. So anyway, this is comes under the autotrophic bacteria. Another one is heterotrophic bacteria. In heterotrophic bacteria, fear, saprophytic, fear, parasite. And also symbiotic type of bacteria also there. In symbiotic nature, the both are get mutual benefits. Is it clear? Hello?
Yes, sir.
It is important.
Yes, important. Nutrition, bacterial nutrition also important. In bacteria, each and every part important. Okay?
In bacteria, sir?
Bacteria cell structure important, nutrition growth important, reproduction important, and economic importance. These are all.
Okay.
Right? We will continue next class. I will complete about the virus and cyanobacteria and fungi. And fungi also. Okay?
Sir, excuse me, sir.
Ah.
Sir, if possible, can you give the important questions?
I will give my last class.
Okay, sir. Thank you.
Okay,
Difficult
right.
to learn.
What?
In exams, what it is coming that?
In exams?
It's difficult to learn, sir, if you give in last class. If possible,
At
just
least
give it.
passing marks, sir.
When will you exam?
Exam is on from 28 it is starting, sir.
So actually, how many blocks in your book, in your course?
4 blocks, sir.
There are four
4
blocks
blocks.
are there, sir.
Before I giving to the important question, just you select any 2 easy block out of 4.
So what you have explained from that only you can give all.
That's also enough. Actually, so far I explained
Could we
about the
go
bacteria, complete bacteria. That means black 2. In
block
black 2
yes,
unit
sir.
completed. Somewhat unit 4 also completed. That means while the comparison between bacteria virus, some portion also have completed in virus. In my next class, I will complete virus and cyanobacteria. If I complete virus and cyanobacteria, complete second block finished. And already the third block started. I'll get general character.
Hmm.
When I was explaining about the general character, clearly told you, just focus on general character. Based on general character, you can understand, you can read 8th unit, that is life history of algae.
Yes, sir.
I have completed only 7th unit, general account of algae.
Yes, sir.
On that class, I clearly tell you, if you get the concept, that means understand about this general account of algae, you can easily understand about 8th unit. And that day I have clearly explained, no need to explain life history of algae.
Yes, sir.
I
Yes.
told you, I suggest you, you can read yourself. If you have any doubt, ask me. I will explain my further classes. So far, no one asked me.
Sir, in algae,
Yes,
3 life cycles are there.
yes.
Which is the most important, sir?
What are those 3 important?
Like Saragam, Polysophonia.
Hmm.
Which life cycle is most important, sir?
How
If
would
you
I
have
know?
to
They are all
comment.
important. They are all important. That's why I told you, just focus on the general account. If you know the general account, if they ask Polycythonia or Unogonium, any type of alga, you can draw the life cycle.
Yes.
Okay?
Sir, you are not setting the question paper.
Yes, I am not. I am. But why should I tell?
Important.
And fungi. Fungi also same. Just focus on the general account. Based on the
Fungi.
general account, the life history of fungi would be easy to you.
General account. Okay, sir.
Okay?
Yes.
Right.
Thank you, sir.